Okay, today we're going to discuss the first bacteria belonging to your gram-negative. So we have here the gram-negative bacilli. So we'll be discussing here all about the Enterobacteriaceae, followed here by the Pseudomonas group. We have also the Vibrio group, and we have also your Campylobacter. So basically, these are three genus here for our gram-negative bacilli, and they are differentiated from one another by number one, their oxidase test. For the oxidase test, your Enterobacteriaceae, lahat ng members nito are oxidase negative except your Plasmonas shigelloides. Then we have also here your McConkey. So the McConkey agar will allow the growth of all your family here except your Campylobacter. And we have also in terms of the carbohydrates with the glucose. So your Enterobacteriaceae try to ferment the glucose, whereas your Pseudomonas try to oxidize their glucose. Vibrio also try to ferment the glucose, but your Campylobacter is inert with it's neither ferment nor oxidize your glucose. So we start our discussion with the family of your Enterobacteriaceae. So these are your enterics or your coliforms. So your enterics or coliforms usually try to inhabit our large intestine. So usually they can be recovered in your fecal sample, and they serve here as our contamination of your water supply or even your food supply. Okay, we have your generic characteristics of your Enterobacteriaceae. First one, so again, this is a gram-negative bacilli. Some of them are cocobacilli, just the case, for example, of your Escherichia coli. Then we have also oxidase negative. Okay, then they able to grow in the McConkey agar, and they try to ferment your glucose. Okay, another one, they able to reduce the nitrate to your nitrite because they have an enzyme, nitrate reductase. The another one, all are motile at room temperature except your Shigella, your Klebsiella, and even your Yersinia pestis. Okay, we have here some of the general classification of our members of our Enterobacteriaceae. So, we have that one, lactose fermenter. So, ang lactose fermenter lang natin, most likely we have here your Klebsiella, Enterobacter, and we have also here your Klebsiella, wait lang, Klebsiella, Escherichia, Serratia, ganun. Salmonella, Shigella, okay, and Proteus are most likely they are non-lactose fermenter. All the rest are lactose fermenter. Salmonella, Shigella, Proteus are non-lactose fermenter. All the rest are lactose fermenter. Then we have here your hydrogen sulfide positive. So hydrogen sulfide, they try to produce here the H2S, and that is characterized here by the blackening of the colony. H2S most likely produced here by your Salmonella, except your Salmonella paratyphi, which is not producing H2S. We have also here the members of your Proteus. All the species are producing H2S, including also your Morganella, your Providentia. Those are your proteic group, Proteus, Morganella, Providentia. They're also H2S. Ay, no pala, Proteus lang pala. Sorry. Proteus lang, that's your H2S positive, and plus your citrobacter from the eye also H2S positive. Okay, for the indole positive, most likely you could have here your Escherichia and Proteus. And for the race positive, we include all the members ng ating proteic group, Proteus, Morganella, and Providentia. They are your race positive. Okay, we'll start our species of your Enterobacteriaceae with the opportunistic pathogens. When we speak about opportunistic pathogen, they try to cause infection for those patients who has been immunocompromised. Okay, so they are opportunistic pathogens. So first one is Escherichia coli, try to cause a variety of infections, including your urinary tract infection, diarrheal disease, your septicemia, and even some CNS manifestation. On the first one, we have here the strain uropathogenic E. coli. The uropathogenic E. coli, uro means to say urogenital, so I try to cause here urinary tract infection. Okay, for that, we have here different variance factors utilized by the uropathogenic E. coli. First one, we have here the pili that serve as its attachment organ. In order for that to attach, to your urogenital tract and try to cause severe damage or infection. Cytolysine is your hemolysin. So this one try to primarily inhibit the immune effector cells function. Immune effector cells here, like for example, specifically your phagocytes, which try to inhibit the chemotaxis. Chemotaxis is the migration of your phagocyte towards the tissue injury. Okay, another one, we have your aerobactin. So the aerobactin try to chelate the iron component, which try to deprive the host here with the iron as its growth requirement. Again, your uropathogenic E. coli try to cause your urinary tract infections. Okay, next we have here is the strain of your E. coli which try to cause your diarrheal disease. The first one, we have the ETEC or your enterotoxigenic E. coli. Enterotoxigenic E. coli try to cause your traveler's diarrhea. So, equivalent na traveler's diarrhea ng Giardia natin. Only that one is a parasite, but this one is your bacteria. Also called here your Montezoma's revenge or Turista fever, ganun. Okay, so we have here the variance factor why try to cause here diarrhea. So the first one, we have here the fimbriae. The fimbriae serve as its appendage, allowing this ETEC to bind to your gastrointestinal tract, epithelial tissue, and try to cause disease, and then try to release the toxin that try to cause here diarrhea. The toxin, it try to produce here dalawa, so we have your heat stable and heat labile na toxin. Okay, so the heat labile toxin, again produced by the ETEC or enterotoxigenic E. coli, has two subunits or two fragments. Again, always the fragment A is the one that try to cause the cytotoxicity sa lahat ng mga toxin natin. And always the second fragment or the fragment B for binding, so that one responsible here for allowing the toxin to mediate or allowing it to bind to your tissue in order for that to cause a damage. So, magbabind mo na siya. So, kailangan ng fragment B. And then, once it bind, ang nagkakos ng damage dito or effect or cytotoxicity would be the fragment A. Okay, so it try to cause here your traveler's diarrhea basically because of, again, this toxin try to increase your adenylate cyclase enzyme. It try to increase your cyclic AMP, which eventually results to your hypersecretion of the water in your intestinal tract and electrolytes, and nagkakaroon tayo ng diarrhea. But the diarrhea here is purely diarrhea lang, the watery diarrhea, walang blood, walang mucus, walang pasas. On next one, we have your AIAIC or your enteroinvasive E. coli. Enteroinvasive E. coli here resembles our Shigella toxin. This is your Shigella-like infection. This is differentiated from your Shigella because the diarrhea produced by your AIAIC enteroinvasive is actually watery. Meron siyang blood pero wala siyang mucus. Just like ang Shigella natin, meron siyang watery diarrhea, may blood, may mucus, may pasas. But your enteroinvasive, that one would have been characterized by wala siyang pus and wala siyang mucus. May blood lang and watery diarrhea. Okay, another one, we have also here identification, differentiating this one as to your non-lactose fermenter. Remember, your E. coli is a lactose fermenter. Siya is lactose non-fermenter. Another one, non-motile siya. By virtue of the characteristic ng E. coli natin, E. coli is a motile, pero siya is non-motile. Okay, another test for identification of your AIAIC would be the Serenity test. This is your animal inoculation where you try to get a sample from your patient, inoculated one to your guinea pig, and then try to observe for the kerate conjunctivitis. Para siyang sore eyes, ganun namumula. Pag nagkaroon siya ng kerate conjunctivitis, so we consider that one as positive, mean to say, the one you have isolated from your clinical specimen is your AIAIC na strain ng ating E. coli. The next one, we have your enteropathogenic E. coli or your EPEC. This one try to cause your nursery outbreak or the infantile diarrhea. Okay, the diarrhea characteristics of your EPEC is actually watery diarrhea. Meron siyang mucus, meron siyang pus, pero wala siyang blood. Okay, the next strain, we have your enterohemorrhagic or EHEC. Enterohemorrhagic E. coli. So this is your 015787 na strain. Okay, so enterohemorrhagic E. coli, other name for that is your verotoxic. verotoxic E. coli because it's been identified to cause severe damage with the vero cell of your African monkeys. Okay, another one, it tried to cause here, basically, itong strain niya causing here hemorrhagic colitis. Colitis is your colon, di ba, inflammation ng colon. Hemorrhagic, we're expecting that one na may blood. Another one, we have your HUS, hemolytic uremic syndrome. And then it tried to produce here to toxin. We have the toxin 1 and your verotoxin 1, verotoxin 2. The verotoxin 1 resembles also your Shigella toxin or Shiga toxin produced by your Shigella dysentery. Okay, so basically that one tried to produce also diarrhea just like your enteroinvasive, di ba? Parang Shigella toxin din siya. This one also produces a watery diarrhea with blood, kaya hemorrhagic ito. But then again, this would have the absence of the pus or the absence of your mucus. Okay, identification of your EHEC. Number 1, we have your SMAC test or your sorbitol McConkey test. Usually sa McConkey natin, we are using lactose. Okay, but this one in your SMAC, sorbitol ang ginamit natin to check if your organism here try to ferment the sorbitol or hindi. In the case of your EHEC, hindi siya nagpa-ferment ng sorbitol, giving this one a colorless, the colony characteristic in this culture media, and we consider that one as negative, and that will identify your EHEC. Your MAG, another test, we have your MAG, your 4-methyl umbiliferyl beta-glucuronide for identification of the enzyme beta-glucuronidase. Okay, positive result for that would have here the fluorescence. Pag walang fluorescence, we consider it was negative, and your EHEC is negative for the MAG. Okay, these two tests eventually will identify your EHEC. Okay, then we have here your enteroadhesive E. coli. Enteroadhesive E. coli would have here two. One is your diffusely adherent E. coli. Okay, so basically that one try to cause here your urinary tract infections and diarrhea. Then we have your enteroaggregative E. coli. So aggregative because it tried to bind to your cells. Okay, cells here, human epithelial cells is a type of cell line. Pag nag-bind siya doon, nag-perform siya ng mga stacks, parang alternate, parang ganun, stacks, stack of brick arrangement. But this one is only causing here diarrhea. Hindi siya nagkakos ng urinary tract infection. Okay, next na opportunistic pathogen natin, we have here your Klebsiella and your citrobacter. Okay, so specifically we have here your Klebsiella pneumoniae. Klebsiella pneumoniae, the name for that is your Friedlander's bacillus. So being pneumoniae, it tries to cause pneumonia or upper respiratory tract infection, wound infection, septicemia, bacteremia, or even your meningitis. Basta lahat sila na nasa opportunistic infections, most likely they are causing the same type of infection. They are nosocomial infection and they are also causing infection here for those mga immunocompromised patient natin. So lahat sila basta opportunistic. It includes their infection like urinary tract infection, respiratory tract infection, wound infection, septicemia, pare-pareho lang sila. Klebsiella pneumoniae is basically that one would have here the capsule. And dahil sa capsule niya, it appears to have here very large na colony and most likely that one is highly mucoid. Again, because of the capsule. It is also associated with your extended spectrum beta-lactamase. So, ito ay mga strain na they are most likely resistant to your antibiotic treatment, especially for the mga penicillin group because of the lactamase, beta-lactamase nila na enzyme. We try to destroy the beta-lactam ring of your antibiotic like your penicillin. Okay, we have here the biochem between your E. coli, Klebsiella, and we have also here your Enterobacter. Because almost they belong to the same naman na group. Klebsiella and citrobacter, Enterobacter, most likely. So we have the summary of that. We have here the IMVIC reaction. Pag sinabing IMVIC, that's your I stand for your indole, M for your metal red, and V for the Voges-Pascauer, and your C stand for the test for your citrate. So pag sinabi natin E. coli, ang kanyang IMVIC reaction is positive, positive. Okay, that's your positive, positive, negative, negative. Pag sinabi positive, positive, Okay, positive is the first na test for the eye, for the test for the eye or the indole. Okay, the next positive is also for the next test, which is your metal red. So, ganun siya. Again, your E. coli is positive, positive, negative, negative. Your Klebsiella pneumonia, subspecies pneumonia would have your negative, negative, positive, positive. Your Oxytoca would have your positive, positive, negative, positive. And for our Enterobacter, so it would have a negative, negative, positive, positive. Enterobacter and your Klebsiella pneumoniae would have your different na impic reaction. So differentiate the two kung ano ba talaga. Then you perform your other tests like your lysine decarboxylase na test, ornithine decarboxylase, and we have also here your arginine dihydrolase na test. Okay, for the lysine decarboxylase, it will give a positive results for all your Klebsiella, including your pneumonia and your Oxytoca, plus your Enterobacter erogenous, but not in your Enterobacter cloacae. Okay, so that one will have here a negative result for that. Ay, sorry. That will have your negative na result. Para ma-confuse. Okay. Again, your lysine carboxylase, it will give all the positive result here with your Klebsiella, for both your Klebsiella pneumoniae, Klebsiella oxytoca, and even your Enterobacter erogenous. But not with your cloacae, it will give a negative results. Okay, for the arginine, for the ornithine decarboxylase, this test will give a positive result sa dalawang Enterobacter natin. Erogenous and cloacae lang ang positive. Arginine dihydrolase will only give a positive result with your Enterobacter cloacae. Okay, so by this test, it will help us differentiate between your Klebsiella pneumoniae and then Enterobacter because again, they have the same impic reaction. They have this negative, negative, positive, positive, the impic reaction. Okay, the next one, we have your Serratia and then your Citrobacter. So, since they are also opportunistic, they try to cause the same infection, UTI, upper respiratory tract infection, wound infection, and we have also here your septicemia. Okay, so they are ONPG positive for both your Serratia, lahat ng species ng Serratia natin, and even your Citrobacter. Okay, ONPG stand for your orthonitrophenylgalactopyrenoside. So this is the test for lactose fermentation. So they are lactose fermenter. Okay, so basically we have here Serratia marcescens produces here a very characteristic na colony pigment, very red na pigment, and that is responsible here for the pigment would be the prodigiosin. Okay, this is also positive for the lipase, gelatinase, and dinase. Okay, so dinase natin pareho sa ating staph aureus, positive din siya sa dinase. Then we have also here the Serratia odorifera. So from the words of odorifera, so medyo mabaho ito, it would resemble here your musty odor like your rotten tomatoes. Okay, so there are two biogroup. The biogroup 1 is isolated from your upper respiratory tract na specimen, and that will be identified here being positive to your Rapinus, Arabinus, and your ornithine. The biogroup 2 ng ating Serratia odorifera, this one is isolated from your CSF and even from your blood. Okay, this one is identified being negative for your Rapinus, Arabinus, and even your ornithine. Okay, we have here the biochem reaction of your Serratia, and then we have also your Citrobacter. Serratia marcescens, Citrobacter frontii, and we have your Citrobacter cosseri or your diversus. Okay, TSI, triple sugar iron agar, so that's A over A. Again, A is your yellow na color sa taas. Mean to say, try to ferment your glucose, sucrose and lactose. Ang sa baba natin, di ba, that's your glucose. So, try to ferment, then kaya yellow, ito kaya naging A. Okay, this one, K over A. Gas formation is identified by the crack in your colony, and that will be identified sa lahat naman. But your H2S, water talaga. Okay, H2S or hydrogen sulfide, blackening of the colony, can only be identified with your Citrobacter frontii lang. We have here the impic reaction. So again, the step for the indole, metal red, phosphorus in the citrate. The in vivo reaction of your Serratia marcescens would be your negative, positive, positive. So, negative for the indole, lahat metal red, Vox-Pascauer, and citrate will give a positive result. For citrobacter fungi, it would have your negative, positive, negative, positive. And then for your citrobacter coceri, it will have your positive, positive, negative, positive. Then we have your KCN, that's your potassium dichromate. So, pag nilagyan mong culture media ng potassium dichromate, potassium cyanate pala. Okay, pag nilagyan mo ng potassium cyanate and culture media natin, mag-grow ang ating Serratia marcescens and your citrobacter fungi. Ibig sabihin yan, they tend to tolerate the presence of your potassium cyanate. Unlike this one, your citrobacter coceri, it will not grow in the presence of your potassium cyanate. Okay, the next opportunistic pathogen belonging to your Enterobacteriaceae, we have here the Proteae group. Okay, or the family that includes your Proteus, Morganella, and we have also your Providentia. Just like your other na mga opportunistic pathogen na Enterobacteriaceae, they're also causing here upper respiratory tract infection, urinary tract infection, septicemia, bacteremia, wound infection. Okay, so this group here identified as being urase positive. So lahat sila urase positive. Another unique to them, they are phenylalanine deaminase positive. Okay, in the case of your Proteus, so this is capable, so of all the three here, only the Proteus is capable of the swarming motility. Although all of them here are capable of motility. They are motile. Okay, so again, ang hindi natin motile, Klebsiella, and we have also here your Shigella, Klebsiella, okay? And even your, your Aiahic, interoinvasive na E. coli is also non-motile. This one is motile, so all of them are motile, only that your Proteus is capable of swarming motility. Swarming motility, nagsaswarm sa ating, parang siyang parang may transparent na haze, ganon. Para siyang may parang may may fogging, ganon, sa culture media mo, the swarming, parang gumagalaw. Okay, the swarming motility here is actually a regulated cycle of the swimmer and the swarmer na cell. The swimmer cell here is the vegetative na form. The swarmer, on the other hand, is the hyperflagellated, elongated polyploid cells, which are capable of the coordinated movement that will be responsible here for the swarming motility. Another thing about that one, this also try to produce here burnt chocolate odor in the swarming capability niya, and that is in the SBA or your sheep blood agar. Okay, for the biochem reaction here, different species of your proteic group. So we have your Proteus mirabilis, Proteus vulgaris. We have also here Providentia, Stewartii, Providentia red jerry, and we have also your Morganella, Morganii. For the in vivo reaction, so in the case of your Proteus mirabilis, is only negative for your indole, or the rest are already positive. Sa atin naman, Morganella morganii, negative lang siya sa citrate. Itong pinakalas na test. Okay, then we have your urase test. Again, urase test will identify the group of this Enterobacteriaceae natin. All of them are positive for the urase. And also, all of them are positive for your phenylalanine deaminase. Another one, H2S, with the blackening of the colony, could only be identified with your Proteus na dalawa. Proteus mirabilis and your Proteus vulgaris, but not with your Providentia, not with your Morganella. Okay, then we have here motility. All of them are motile. Again, only that ang dalawang Proteus, they have the swarming motility. Okay, the next one, we have your intestinal pathogen. So this Enterobacteriaceae try to cause here diarrhea, most likely. Okay, so their activity is more of the intestinal tract natin. The first one, we have your Salmonella, Enterica, subspecies Enterica, serovar. These are the serovariety. Serovariety, cholera, Swiss, serovariety, paratyphi. Parang mali yung spelling ko kanina ng paratyphi. We have also here your serovar na typhi. So again, they are identified by the following biochemical reactions. So for the citrate, okay, so negative ang ating, most likely ito lang magpa-positive. For the gas, napoproduce lang ng gas natin is your cholera suis. Okay, for the H2S with your blackening of the colony, all of them are capable of blackening of the colony except ito, wala siyang para-type P, wala siyang H2S gas. And lysine carboxylase also will identify your para-type P kasi siya lang ang negative. And ornithine carboxylase, ang negative lang natin would be your serovar na type P. Okay, then we have also here the virulence factor associated with your Salmonella, Enterica subspecies Enterica. So they have the fimbriae. Okay, the fimbriae allow this bacteria to attach to your intestinal mucosa in order for that to cause your dysentery, your gastroenteritis, or your food poisoning. Another one, it produces also here your enterotoxin. So, enterotoxin, pag entero, nasa intestinal tract natin siya. So, it try to cause here your food poisoning or gastroenteritis. So, we have here the clinical manifestation or the diseases associated with your salmonella. First one, food poisoning because of your enterotoxin or your acute gastroenteritis. We have also here your enteric fever, simply your typhoid fever. Pag typhoid fever, characterized by very high na fever, pero ang WBC count ni patient natin ay mababa or mababa. Usually, pag may infection ng patient natin, ang WBC count natin ay mataas dapat. Considering na sobrang taas ang lagnat ni patient. But in the case of your typhoid fever, mababa ang kanyang WBC count. Okay, that will identify that one. So, this is caused here by your Salmonella paratyphi and your Salmonella typhi. Ang specimen for that blood, you can recover the specimen during the first and second week of infection. You could also use the stool sample where you can recover the microorganism on the second and third week of the infection. You can use the urine sample where you can recover the specimen on the third and the fourth week of the infection. Okay, then we have here the bacteremia. So it's being caused here by the three species of your serovariety, Salmonella, Enterica subspecies Enterica, serovariety, cholera suis, subvariety, paratyphi, and serovariety na typhi natin. They're causing all bacteremia. Again, you get infected by Salmonella by ingestion of your poultry products, milk products, or even sa mga chicken. I think sa mga wet market, they can also be because of your Salmonella poisoning. Okay, we have your characteristics. So, in the bismuth sulfide na agar, your Salmonella would appear to be a metallic na colony with black na ring. Another one, we have here the test. For identification of your Salmonella, we have your Widal's test. Although yung Widal's test natin is already an obsolete one, so we are doing the Typhidat. Typhidat ang ginagamit natin ngayon to identify your... Okay, Typhidat is for the antibody, IgM, saka IgG na antibody. Madidetect natin, serological test. Although this one is a serological test din naman, but then, okay, this test, if you are using this one, it can give some variety of information based on the titer. Okay, pag nag-positive siya sa O antigen at the titer more than 160, 1:160, then that would signify you have an acute infection or a present infection or acute infection. Currently, you have an infection, ganon. H na antigen with a titer 1:160 identifies here your past infection. You have recovered already. You have been infected in the past. And the VI antigen, positive ka doon with a titer more than 1:160, it would signify that the patient would have a chronic carrier. Remember the patient when na-infection nagkaroon ng salmonella infection, typhoid fever, if the patient has not been adequately treated, the bacteria can stay in the gallbladder of the patient or eventually become a chronic carrier, where every time the patient is shedding a fecal sample, it contains the bacteria, and if that fecal sample gains entry to the bodies of water or source of the water, drinking water in a community, then you can get infected by that. And that you get infected by salmonella by ingestion or, ingestion. Okay, the next one, intestinal pathogen with our Enterobacteriaceae, we have here your Shigella. So your Shigella here is differentiated basically from your Salmonella by the following biochemical characteristics. Shigella is non-motile, Salmonella is motile. And then, except, H2S with your blackening of the colony will give a positive result with the salmonella except your serovar na partyphi, salmonella partyphi. Okay, so Shigella will have your negative result with your H2S. Then lysine carboxylase is positive for salmonella but not your Shigella. We have here your infective dose, how much of the inoculum ang organism natin that will enter your body before you can get infection. So you need to have your 10 to the 6. Ito 100 to 200. Konti lang kailangan mo dito para ma-infect ang patient. Meaning to say this one is more virulent compared with your salmonella. Because a little inoculum here, 100 to 200, you can get already infected. But this one, you need to have 10 to 6 na inoculum size in order for you to get infected. Okay, we have here the serogroup or serovariety of your Shigella. We have here your serogroup A, represents your Shigella dysentery. The B na serogroup is your Shigella flexneri. Boydii I for the C, and we have also here your Sonei for the serogroup D. So again, for the mannitol, all of them are positive for that, except your Shigella dysentery. O and PG positive, again, this is for the lactose fermentation, orthonitrophenyl galactopyrenoside. For the lactose fermentation, we only identify sursonate. Siya lang magpa-positive doon. Okay, then we have here the virulence factors associated with our Shigella. So basically contains also here your adhesin, your fimbriae for attachment to your intestinal mucosa. Another one also contains here the toxins. So in the case of your Shigella dysentery, that one try to cause here your dysentery, diba? So it try to cause here bacillary dysentery or diarrhea characterized by the presence of watery diarrhea, but that one would have here the blood, the presence of the pus, and the presence of your mucus. Okay, so that one produces here two toxins, the neurotoxin and the enterotoxin. All the other na masero group natin, your flexneri, boydii, and your sonay do not have here the neurotoxin, but they have only enterotoxin lang. Okay, so bacillary dysentery caused here by Shigella dysentery produces here watery diarrhea and eventually magkakaroon ka ng blood and the mucus or pus cells. The third one, Enterobacteriaciae, will try to cause here intestinal tract infection. We have the Yersinia. The first one, we have Yersinia pestis. Yersinia pestis is the causative agent ng plague na nangyari nung mga dating panahon na madaming namatay. So that one is transmitted by the bite of the fleas coming from the rodents or rats. Okay, there are different forms of your plague. We have here the bubonic plague or the glandular plague. So, meron siyang enlargement ng glands na the lymph nodes. So, at the site of the bite, karoon ng fever ang patients, which eventually later on, magde-develop siya ng swelling ng ating lymph nodes. Magkakaroon siya parang bukol, inflammation of that swelling that you call it bulbous. Another one, septicemia na plague. So, if the bacteria try to gain entry to your blood circulation, it try to cause your sepsis or septicemia. On the other hand, if that one migrate up to your pulmonary tract, respiratory tract, it become here your pneumonic type of your plague. Most likely, if that one is transmitted by ingestion, or by inhalation of the fleas. Pwede din bite, pag pumunta lang siya doon sa taas. Pero pag in-inhale mga fleas, automatic magiging pneumonic agad siya. Okay, then we have here the lab test for identification of your Yersinia pestis. So that one would have here in the methylene blue or Wasons, it would have here the bipolar staining. Mas sinabing bipolar staining, so kung itong cell natin, dito na part is stained, and dito na part din is stained. Both end would have here this dark staining part, and the other part is hindi sila light. They are light staining on the other part. That gives you here the characteristic na safety pin appearance. Okay, so in the SBA sheep blood agar for 48 hours, it would have here the characteristic na cauliflower na colony characteristic. And then, in your broth medium here, would have your stalactite. I think you're familiar with that. Stalactite, stalagmite. Sa mga cave natin, di ba? Then preferential growth, you're able to grow at 25 to 30 degrees Celsius, but this one is non-motile, hindi siya motile. Next, we have your Yersinia enterocolitica. So they are usually found inhabiting here our domestic animals, including your dogs. Okay, infection is by ingestion of contaminated food, especially the pork. Okay, so manifestation of the disease, enterocolitica, so gastroenteritis ito, acute enteritis. So that's more diarrhea with appendicitis-like manifestation, sobrang sakit ng tiyan ni patient sa tagiliran ni patient natin. Another one, it would have your arthritis manifestation and erythema nodosum. Laboratory diagnosis, so same with your Yersinia pestis, may bipolar staining siya. This one will give a bullseye appearance in your CIN, your cefsolidine ergasan novobiosine na agar, and able to grow here preferentially at 25 to 30 degrees Celsius. And then this one would be motile at 25 degrees Celsius. Okay, then we have also here the Yersinia pseudotuberculosis. Yersinia pseudotuberculosis, just like our Yersinia pestis, is also the disease of your rodents. You could get infected here with a close contact with the contaminated na or infected na animals, or even by ingestion of contaminated na food. So this is manifestation. So once enter your body, this bacteria try to spread. Okay, try to spread to your mesenteric lymph nodes and try to cause your lymphadenitis. Try also to cause your septicemia, and we have also your pseudo-tubercle formation. So parang siyang tuberculosis-like with a plague-like manifestation in your lungs. Okay, we have the lab results. So they're also capable of your bipolar staining. Motile din siya, only at 18 to 20 degrees Celsius. Okay, we have here the biochemical reactions of your three species of your Yersinia. So, TSI, we have your yellow-orange for both, babasa ka sa taas. Christensen's urea would identify the enzyme urease. It will give a positive result only for both your enterocolitica and your pseudotuberculosis, but not in your pestis. Motile natin ay dalawa. So enterocolitica at 25 degrees Celsius, as well as 18 to 20 degrees Celsius. But your Yersinia pestis would be non-motile for that. ODC and Socracy will only give a positive result with your Yersinia enterocolitica. So we have here the CIN. The positive natin would be your pink na colony. So they are positive for the CIN. Specifically with the bullseye colony with your Yersinia enterocolitica. Okay, the antibiotic na cefsolidine, so itong component niya, cefsolidine, it inhibit the gram-positive and even your gram-negative cocci. Gram-positive and gram-negative, novobiosine also try to inhibit the gram-positive na cocci, because our violet try to inhibit here the gram-negative. All the other gram-negative except your Enterobacteria, except your Yersinia, I mean. Okay, so this is all for our Enterobacteriaceae.